In this video, I would like to make a brief introduction of DNA and RNA structures and their conformations. DNA and RNA are both nucleic acids. They carry gene information and regulate protein production. DNA is double-stranded, with B form being the dominant form. This is what Watson Crick found and made them Nobel laureate. The commonly seen cartoon version highlights phosphate groups since they form the very well-known double helix structure and the base pairs sticking the two chains together with hydrogen bonds. RNA, on the other hand, is single-stranded most of the time, with A form being the dominant form. Both DNA and RNA are chains with repetitive units, and a single unit consisting all three building blocks is called a nucleotide. A chain extension can be considered as adding these nucleotides towards a 3' end. Same-wise, dinucleotides and tetranucleotides are 2 or 4 consecutive nucleotides that are built for modeling and parameterization purposes. The movie here is showing a typical tetranucleotide. Now that I've extracted a nucleotide with atomic assignments and dihedral angle notations, whether DNA or RNA prefer A or B or sometimes even other forms depends on the collection of the backbone torsion variables. Since the backbone are indeed constructed via the phosphor-diester linkages, the term backbone is interchangeably used with phosphate. They are the first building block of nucleic acids. One major structural difference between DNA and RNA is the sugaring. DNA with a deoxyribose has a hydrogen atom on C2' position, while RNA with a ribose has a hydroxyl group on C2' position. This is exactly how the two terms DNA or deoxyribonucleic acid and RNA or ribonucleic acid came from. Sugaring is thus the second building block of nucleic acids. The possible choice of bases for DNA and RNA are also different. Only adenine, guanine, cytosine, and thymine are present in DNA, and only adenine, guanine, cytosine, and uracil are present in RNA. Now that we have a full picture of the DNA and RNA structure, we'd like to define their conformation by the dihedral angles noted in the graph. The torsion angle chi corresponds to the glycosidic bond connecting the base covalently to the sugaring. The preferred angle may vary depending on the type of the base and the sugaring conformation, or else known as sugar puckering. Two major conformations can be identified by the dihedral angles between C2' and C3' on the ring. The relative population of C3' endo and C2' endo are close to 50-50 from experimental observation, with variations on the base it attached to. The backbone torsions were also defined as noted in this graph. They're alpha, beta, gamma, delta, epsilon, and zeta. These are the histograms of the backbone dihedral angle distributions collected from crystal structures. In the next video, I will dive into these dihedral angles and discuss how force field parameters were optimized in order to reproduce the experimental measurables, such as 3J coupling. Thanks for watching.